Okay, hello and assalamualaikum. My name is Shazli Iman, and today I'm going to present about my project. So, firstly, I want to introduce which is my group members, group 13. So, today, me and my friends already did our research about our finding a gene project in bioinformatics, bioinformatics 1, and uh, my friends Zakwan, Hamza, Danish, and also me myself going to present about our genes that we find and after we have done our research about tumor protein. So tumor protein, as we can see, tumor is the deadliest things that everyone have in their body, which is not activate or not, also not reactivate. So we're going to dig through about tumor protein first. So what is a tumor protein? So tumor protein is what we call TP53. So TP means tumor protein 53, and also in English, maybe in other words, they're known as P53, which is protein 53. So what is a tumor protein? So it is called due to the mass of molecular mass. It has a 53 kilodalton and also a fraction of cells and proteins. It also the protein also locate at the 17th of chromosomes, and the protein is a phosphoroprotein made of 393 amino acids. So it is called tumor protein because it's a tumor suppressor, which means it regulates the cell division, keeping cells from growing and dividing into rapidly fast in the cells, which is very rapid. So why do we call it a suppressor? Which is suppressor is the guardian of genome, which is because of this role, conserving stability by preventing genome mutation from spreading inside cells. Uh, the genome also protects from before it was emerged the tumor. So the next one is what is a tumor? So tumor normally have inside everyone's body that controls the cells of growth and also division. So it create a lot of older one. It create all the all the cells into a new cells, which is it always mutate and mutate and mutate and so on. So the cells that are damaged are no longer needed, die to make a room of healthy replacement. So it's very important. So what is the difference between tumor and tumor? So tumor also spelled as tumor is also a neoplasma, and as a mass of abnormal tissue arises with arises with without obvious cause of pre-existing body cells. So the next part is I'm going to show you our our project that we did on this protein. So first we go to the NCBI, which is known as National Library of Medicine, uh, which mean National National Center of Biotechnology Information, which is there's a tons of genes, proteins, about cells, and also all the database that have been compiled into one website from the whole country. I mean worldwide. So let's go, which is tumor protein is a gene, which is we finding a gene project. So we just type in on TP53. After we type in on the TP53, take some time to load. So the first you can see is the tumor protein, which is Homo sapiens humans. Also, and then there's a reference. There's a tons of information that you can see, which is you can see it is located at chromosome 17. So the NC is a NC 000017.11 is an example of accession number on certain cases. So let's dive into the first one, which is tumor protein P53. So quite take some times. So right now, alright, as you can see, I need to widen my screens so that you can see this is the outcome after I click it on the TP. I mean, you can see this. Let me widen this first. So as you can see, after we click on the search button, we can see the outcome of the results, which is the TP53. And now we just click it to see the accession number. Just click the left side proteins, and also it will provide a page. So we click on the first one. If you want to run to the blast, which is my friend will conduct that. So this is an example of FASTA. This is a FASTA, which is FASTA is a format which begins with a single line description followed by lines of sequence data. So as you can see, the accession number that we are going to take into the BLAST software is this one, the NP underscore 00537. And also there's a lot of reference and articles about tumor. And the next part, which is my friend will tell about more deeply into the investigating the genes. So this is a type of, this is a reference that you can see most of the time, and importantly is the accession number, so that we can see the result, and also we can examine through the graphic summary and also testimony, or and also the sequence alignment. So thank you.
Assalamualaikum. My name is Amzar Razik Khan bin Azamhan, and for the next step, we will perform a blast search against a DNA database using our desired protein, which is the tumor protein TP53 for the Homo sapiens. First, we search for NCBI. Click the blast link. We will be using T+N. Enter the our desired protein accession number, which is 537.3. Our database, we use express sequence tags, or EST, and organism is nematodes, tag 6231. So perform the blast search. Take a few seconds. Okay, so this is the summary of our protein. Description: we have only one sequence, which we will be using throughout the our project. This is the graph summary, and this is the alignment. So that's it for my part. Thank you.
Assalamualaikum dan salam sejahtera. My name is Muhammad Zakwan Zakirin bin Abdul Razak. Now I'm going to present about information about novel protein in our tumor protein, which is TP53. The novel protein that we found is Trichostrongylis species, which also can be called nematodes or roundworm, which are the ubiquitous among herbivore, including cattle, sheep, donkey, goats, deer, and rabbit. At least Trichostrongylis species have been associated with human infection that occur in larvae that from contaminated vegetable or water. These are the sequence protein of the Trichostrongylis, that we can get from the question 2, which is from the sequence alignment of cellular tumor antigen p53 isoform. The name of the novel protein is Trichostrongylus globin, which is the species is Trichostrongylus vitivinus. And also the lineage of the protein is cellular organism, eukaryote, Opisthokonta, Metazoa, Eumetozoa, Bilateria, Protostomia, Exozoa, Nematoda, Chromadoria, Strongyloida, Trichostrongyloida, Trichostrongylidae, and Trichostrongylus. Next, we're gonna do the blast P for the Trichostrongylus vitrinous protein. We can copy this. We copy this sequence and we paste it to the blast P, and we blast it using blast P. And this is the result of the Trichostrongylus vitrinous protein. So this is the decision of the protein, which is the greatest is p53 domain containing protein Haemonchus contortus, which is the score that this protein get is 176 bits, and the aspect value of 3E-52. The identity is 99 over 121, which is 82%. The positive is 105 over 131, which is 86%, and the gap is 4 over 131, which is 3%. And this is the graphic summary of the Trichostrongylus vitrinous protein. And this is the taxonomy of this Trichostrongylus protein. I think that's all from me. Thank you.
Okay, we continue with number 5. We need create a multiple sequence element with our novel protein, our original query protein, and a group of other members of its family. Okay, so I will show you how we get the multiple sequence alignment. So this is the result, all the multiple sequence element that we get from NCBI website, that we search for our protein, and we need to click on alignment tab. So they will show all the result here for the multiple sequence element. So we search on EBI website using MAFFT. So we use cluster format. So this is all the result that shows here. Yeah. So we continue with number 6. So we need a create a phylogenetic tree using bootstrap or tree routing are optional. We use any program such as Mega, Plot, or Flip. So we use Mega software. So we show how I get this tree result, this phylogenetic tree. So we need to open a file that we need to open Mega file, this Mega file that we create. So it will show the alignment. So this is actually the last step. When we click this alignment, this align using the muscle algorithm, we use the muscle algorithm, and this all the result here. And after that we need to click phylogeny, and we need to click on construct test neighbor joining tree. So after that we will get this three result that we need to click on bootstrap. So that's all from me. Thank you.